Namaskar and welcome to StudyIQ. I am your friend Rahul Saigaonker. Now, before we begin today's discussion, there is a small message from my side to all the aspirants who are appearing for mains examination 2023. I hope that your preparation is going as per schedule. You have very less time left now, a little more than 2 weeks. And as the date for the examination approaches, students, aspirants, they tend to get a little jittery, they tend to lose confidence. My advice to you would be to give yourself credit and have faith in yourself. Do not lose that confidence. And that is our intention through these discussions as well. We are trying to help you by discussing some of the relevant topics which will be important for mains examination 2023 from science and technology. There is no need to get overwhelmed here. What we are discussing is something that you already know. We are simply trying to complement or add a little more value to it. As the date approaches, students get a little jittery, they tend to read things or they tend to question themselves. They think that I'm not able to recall anything. That's very normal. That happens with everybody. This is the time not to lose confidence, but to consolidate and revise. Stick to your path. All right. Right. After that message, let's begin today's discussion. In our previous session, we discussed in general about biotechnology and its important applications. Today, we are going to focus on genetic engineering. We'll talk about different ideas, different technologies that have emerged connected to genetics. There are two things I want aspirants to understand from this. First of all, specific applications that we have brought. Apart from that, also understand the evolution or development in this field of genetic engineering. All right? Right, let's begin. But before that, there is a small message for all of you. The August batch is closing out on 31st August 2023. Before that, do enroll. The classes have already begun. Very few classes are over. You can already see the recordings, but this train will be gone. The admissions will be closed by the end of this month. And this is already late. I would say this is appropriate time to join this batch for 2024, because if you delay any further, then 2024 preparation would be a little question mark. Probably for 2025, you need to get ready. So decide within a few days and enroll for our program, because this is the most comprehensive program for UPSC civil services preparation, where we will be with you for prelims, for mains, for interview. A mentor will be allocated to you right from day one. Prelims test series is included in this itself. Mains answer writing program or mains residential program is also included in this. The whole shebang, a comprehensive package at a very affordable price. For max discount, do use my code Rahul Live before 31st August. Enroll. I'll see you in the class. Right? All right. Let's talk about genetic engineering. Now, what is the meaning of genetic engineering? Now, it is a field. First of all, engineering. Engineering means you do build something, you alter something. And what do you alter? You alter the chemistry of genetic material. And what is our genetic material? Our genetic material is this DNA. Now, a locus of this particular DNA is a gene. Now, this gene has a specific characteristic or a trait. And that particular gene, it will lead to encoding of a protein, and that protein has a role in our body. Right? Now, genetic engineering involves altering the chemistry of the fundamental unit of our genetics, that is the DNA. Now, if you look at genetic engineering as a field, it is relatively newer. Although from, I would say, time immemorial, we have been talking about changes in genes through a natural way or within species, changing of genes within the species or adaptation of genes within species, evolution of genes within the species through a concept called hybridization. Now, hybridization always works intra-species. Now, you must have seen this, that say the classic idea of grafting. I take a stem of red rose and I take another stem of, say, white rose, and I graft those two. Right? You'll probably get the development or the branch that evolves that would give you pink flowers. Right? That we have seen the basic idea of hybridization. But hybridization is always intra-species. When you remove this intra and add inter, now this becomes your idea of genetic engineering. That means genes from some other species or some other organisms incorporate into a different organism or a different species. And this field is in vogue or in development since 1980s. I would say since 1980s, it has gained a lot of traction. And today, when we look at 21st century, genetic engineering has so many applications. So many applications. Genetic engineering is applied for developing recombinant DNA. It is used for gene cloning. It is used for gene transfer, stem cell therapy, gene mathematics, gene physics, biophysics, biostatistics, so many things. In fact, genetic engineering is a larger term under which we get therapeutic idea using genes for therapeutics of a treatment. So gene therapy is basically a subset of genetic engineering. Please remember, genetic engineering can be used for good purpose. It can also be used for bad purpose. I hope you do understand. Now, we only talk about good things. Especially under genetic engineering, we talk more and more about gene therapy, where we alter the chemistry of the genes, or we alter the chemistry of the DNA and try to improve treatment, diagnostics, and provide some relief to patients. That is one of the important applications that we have seen from genetic engineering. Now, apart from that, there are so many other applications as I mentioned here. One of the first ideas that evolved through genetic engineering was the idea of recombinant DNA. From 1980s, scientists have been working on developing different kinds of recombinant DNA, which has found so many applications as of today. Under genetic engineering, we first of all learned how to manipulate the cell structure, enucleation, renucleation. Then we try to introduce genes from one organism into another organism. Please remember, when I edit or when I change a DNA within an organism, it is called as a cisgenic change. Cisgenic means where I do not incorporate any new gene. Basically, no new gene or no transgene has come. No new gene from outside. But when I use the term transgenic, that means genes have come from some other organism or genes have come from outside. So recombinant DNA is basically a concept or an idea of developing transgenic organisms. It may be transgenic microorganisms, it may be transgenic plants, or it may be transgenic animals, right? Now, what is this recombinant DNA? That is a technology where we used enzymes to cut and paste DNA sequences of our interest. Now, how do we do it? See, the recombinant DNA technology, it starts first of all with isolation of genetic material of my interest. Say, imagine I know I have studied DNA, and in that DNA, I know a locus of that particular DNA is a gene that encodes, say, growth hormone. Now, what I want to do is I want to create or produce more and more of this growth hormone, because it can be used for therapeutic applications, meaning those who are underweight or say those who are stunted, give them growth hormone. Probably you'll see growth in them. Yes. So that's why I have to isolate the genetic material. From where? From whichever interest organism you need. You understand this is a gene. If I add this gene to one particular organism, growth hormone can be produced. So first I isolate the genetic material, and then I incorporate this genetic material in a vector. In a recombinant DNA technology, we always use a vector, and most prominent vector that we use is a bacterial cell. For example, E. coli. Please remember, a vector can also be a virus. Again, that is from prelims point of view, but a vector cell is needed. Normally, we use bacteria as a vector. Why? Because bacteria itself has a DNA. It is not very advanced cell. It has its own DNA spread throughout the cell. It's a prokaryotic cell, you know. But in a bacteria, there is a plasmid. And this plasmid is independent of the original DNA that the organism has or that the bacteria has. Meaning, when the bacteria reproduces, when the bacteria undergoes binary fission, this plasmid will also undergo binary fission, but it is not dependent completely on this. It's independent. So what do? I take out this plasmid using genetic engineering techniques. I take this plasmid. Now, plasmid basically consists of hardly 1 or 2 genes. If we look at the amount of information that it has in some kbs, okay, when we convert that into the actual digital information, it has 1 or 2 genes. So what I do is I cut a part of this plasmid so that I incorporate the desired gene. From a human cell, I understand that one particular gene is responsible for growth hormone. So I cut this particular gene in the plasmid. I incorporate the growth hormone in the plasmid, and this plasmid is again reintroduced in the bacterial cell. Now, when the bacteria undergoes binary fission, you do understand when favorable conditions are given to the bacteria, it is going to reproduce. And when it reproduces, this DNA, that is the plasmid DNA, whatever it encodes, say it encodes growth hormone, that is also produced by the bacteria continues. So what we get? We get some specific kind of chemical. Here we got growth hormone. Now the question is, sir, I understood the basic process. I know the basic process. How do you cut and how do you paste these things? Now, in initial years from 1970s, 1980s, 1990s, we have been developing, or we have been looking for newer kind of enzymes. Now, for cutting the gene, we use restriction enzymes called endonucleases or exonucleases. Now, using this, specific cut is made in the DNA. And we do understand that I want more and more copies of this, because I cannot rely only on a single piece. So what I need is a polymerase chain reaction. That is also part of genetic engineering. You must have undergone the RT-PCR. Now, in the RT-PCR, what they do, they have taken your small sample, but they have amplified that sample. How? By using a polymerase chain reaction. And for that, we use polymerase enzymes. Now, these are used for synthesizing exact replicas or exact copies of the gene or of the DNA. Now, what we do, we cut the DNA wherever required, we amplify that particular gene, and then for joining our desired genetic material into the plasmid, we use ligation, and this is done through enzymes called ligases. And then ultimately, by using different genetic engineering based tools like the micro injection, electroporation, or gene gun techniques, these are the techniques that are used even for say enucleation or renucleation also. Now the plasmid is reincorporated, or the engineered plasmid is reincorporated in the bacteria, and then the bacteria is cultured. Now, using this, what have we achieved? Recombinant DNA can be used for many things. Recombinant DNA can be used for creating vaccines, for creating chemicals like growth hormones, antibodies, different vector-based research. Recombinant proteins are produced. Apart from that, it is also used for genetic modification. Genetic modification, genetically modified crops, microbes, animals. It is also used for different gene therapeutic use. In fact, I can say recombinant DNA technology is one of the precursors or for understanding the CRISPR technology, clustered regularly interspaced palindromic repeats, that has been in vogue or in news these days. Apart from that, it is also used in many engineering applications, say for biofuels, for bioethanol, biobutanol, etc. Now, question is, sir, you told me, okay, recombinant DNA is something which is in development phase since 1980s, 1990s. We have found out different kinds of, say, different kinds of restriction enzymes, different endonucleases, exonucleases. We have also found out different polymerase enzymes and also ligases or ligation enzymes. Using this, one of the biggest applications that we have brought is the genetically modified crops. Now, I gave you an example here. Now, what have we done is, here we have produced growth hormone. This was an example of growth hormone using recombinant DNA technology. I took out the desired gene here. Then I took the plasmid from the bacteria. I cut a gene wherever this has to be incorporated. I incorporated it here using restriction enzymes. I cut using ligases. I join, and then I incorporate that back in the bacteria. Now, this is the basic recombinant DNA technology. Using this idea itself, we have created different genetically modified crops in our country. How have we done it? See, for instance, if we talk about genetically modified crops in India, we have developed BT cotton, which has already been approved. We had developed BT brinjal. It was not approved. It was disallowed in 2021 after a lot of protests. GM mustard is also developed recently. Dhara mustard hybrid 11 has also been approved in 2022. This has been approved by the Genetic Engineering Appraisal Committee. It is under the Ministry of Environment, Forest and Climate Change, which is the apex approval body for genetically modified crops or organisms in our country. Again, there are representatives from DBT in that, but you need to understand there is one problem with entire biotechnology area which we discussed in the last class. The problem is there is a Department of Biotechnology under Ministry of SNT. Then there is a GEAC under the MOEFCC. You will find that multiple ministries are involved, which leads to some coordination convergence issues. Now you have a question, sir, how did we develop this? For example, if we talk about, say, BT cotton. Now what did I do with BT cotton? With BT cotton, BT stands for a bacteria called as Bacillus thuringiensis. Bacillus thuringiensis. Now what I have done here, I have taken some requisite gene. First thing was isolating the genetic material that I want. Now I took a genetic material or a gene from Bacillus thuringiensis, which is called as CRY1AC. Now this CRY1AC gene, it is responsible for generating the BT toxin, and this BT toxin, it is harmful, or it is going to kill the bollworm which harms the cotton. Correct? So first thing is I have isolated the genetic material. Now you need to understand, sir, why do I create genetically modified crops? Because I want certain benefits. I want say resistance against some pests, resistance against the climatic factors, or I want to improve the productivity. For enhancing the yield, for enhancing the productivity, I bring some kind of new gene which gives me some resistance or some enhancement in the crop. That is why I bring it. Now here I brought this new gene from a bacteria into cotton for protecting it from the bollworm. Now what I did was I isolated the CRY1AC gene. The first step is isolation. Then I need a vector. Now I took a vector also. Now for this Agrobacterium tumefaciens, I took another vector called as Agrobacterium tumefaciens. Now in that I had a plasmid DNA. Now this CRY1AC gene was incorporated as a plasmid or as a new gene here. And what I did was I used this bacteria to be incorporated in cotton cell. I added a new gene. Please try to understand what we have done with BT cotton is we have not edited its DNA. What we have done is we have incorporated one external gene into the cotton cell. That means addition of one more gene. So what happens is this cotton cell, when it is cultured, I get tissue culture, I get saplings. Then slowly and steadily I get that one plant where I get cotton seeds, and by breeding that I have gotten the BT cotton. Now this is the basic idea using recombinant DNA technology. Same idea was also used for GM mustard as well. Now in GM mustard, which has been approved, as I told you, in 2022, the scientists, they have created a hybrid. Please remember, I am not discussing too much about BT cotton because it is an old news, but I might expect some questions on genetically modified mustard for sure. Now this genetically modified mustard, it has been developed by University of Delhi by a team led by Deepak Pentel. What they did was they hybridized two variants of mustard. Now you have a question, sir, why did they use this hybridization technique? Because natural hybridization is very tough in mustard because it is a perfect flower. It pollinates immediately, even by a small movement of wind or by bees, etc. Self-pollination takes place. Cross-pollination becomes very tough for mustard. So that's why what this team did was they took two variants of mustard. One was the Indian Varuna variant and another one was the East European variant. Now what they did was they have incorporated new genes in both these. The genes were taken from a soil bacterium called as Bacillus amyloliquefaciens. From this, a gene called as Basta was taken. From this, a gene called as Barnase was taken. And Barnase gene was incorporated in Indian variant. Now, what this does, it suppresses the male fertility. Means it basically suppresses the anther from the Indian variant. And on the other side, for the East European variant, they have used Barstar to enhance its fertility, the male fertility, the anther, its fertility is enhanced, so that when we cross these two, the hybrid that is produced, it will be fertile, because I want that these seeds can be used again in future as well. So I get a fertile hybrid. So if anybody asks you, sir, what is genetically modified mustard? It is a hybrid. It is a hybrid where the parents, that is the East European variant and Indian variant, they are genetically modified. Apart from that, another third gene was added, which is called a bar gene, which provides resistant to specific herbicide called as phosphoenithricin. Now, this phosphoenithricin is sold by a commercial name called as Basta. All right. So basically, what we have done is I have created a hybrid whose seeds are very fertile. I can use them effectively with good germination, with good yield. Apart from that, I incorporate another gene which gives it resistance from a specific herbicide. So what would be its benefits? If you ask the team led by Deepak Pental, they will tell you that there are so many benefits. The first benefit that the team tells is the yield of this DMH11 is 30% higher than the normal yield. Now, if the yield is higher, if the productivity is higher, that means I can reduce the import of the mustard seeds. I can reduce the import of other oils. So I can go for more and more domestic production. And these are hybrid seeds. That means they are sustainable. As you know that the hybrids are fertile. You can use them in the next generation as well. So it is going to help the farmers. Apart from that, now farmers will save a lot of money in manual effort of weeding because they do not need any manual weeding. Why? Because if you simply apply Basta on this, that means phosphoenithricin, if you apply it, nothing will happen to the mustard, but all the other weeds, they'll die. So imagine the amount of benefit that the farmer gets. Farmers' income would enhance. How? First of all, their input cost is reduced. And the output is already high. So you understand now? So this makes sense. However, whenever we talk about genetically modified crops in India, there has been a lot of protest against them, especially genetically modified foods or crops which have to be consumed or which are consumed. And for that reason, there has been a lot of protest from environmental activists and some right-wing organizations, especially because in this, the East European variant is used and they suggest that DMH11 is not completely Swadeshi. And for that, questions have been raised. The matter went to the apex court and the Honorable Supreme Court is also listening to this. But right now, there have been some positive riders along with the approval that has come from GEAC. The GEAC has said that in the next couple of years, what is going to be the impact on bees that will be studied of this genetically modified mustard? Will it impact the bees? And why I'm talking about bees? Because these are most important pollinators. Now, because of this, a study is to be conducted for the next couple of years and a report would be submitted. Apart from that, there have been some concerns which have been highlighted that because of the genetically modified organisms, genetically modified mustard, will there be any application or will there be any harm on the wild population? Right now, you have created or you have incorporated a gene called as BAR, which gives resistance against this particular chemical. That means if you apply this chemical, the weeds are gone, but mustard will not go. What will happen if this company or this particular brand, if it creates a monopoly? Will it hamper the farmer's interest? Yes, right now the manual weeding efforts are reduced, but will it also lead to unemployment? Will it have any impact on cancer incidences? Will genetically modified mustard create some carcinogenic impacts in future? Moreover, we do not know what is going to happen in the long run. Apart from that, some environmental experts, they also suggest that because of the chemical that you're using, some weeds might become resistant to it. Today you are telling that if I apply this Basta on my GM mustard, nothing will happen to GM mustard, but all the other weeds would go. Imagine tomorrow I apply the same thing, but there are some weeds which are remaining because they have become resistant to this. Then what? Will there be super weeds because of this? These are the questions that have been raised sometimes over genetically modified mustard, but there is a keen eye on this. Let's wait and watch, because approval has been given in 2022 itself. If there is any development, keep a track of this, especially for the candidates who are appearing for 2024 preparation, because yes, from mains perspective, this debate becomes very important for you. What can be the positive impacts, as well as what are the issues concerned with the genetically modified mustard, or in general you can simply say genetically modified crops, their benefits and their issues, just an extension of this particular debate. Clear? So what we discussed was, we understood recombinant DNA, and from recombinant DNA we have seen so many applications, and one of the applications is genetically modified crops, which is quite extensive in our country. Now, if you look at the global scenario, in India, we face a lot of issues. In India, there are a lot of protests connected with genetically modified crops, but at the global level, there are many countries who have approved commercial application or commercial expansion of genetically modified crops. In the South Asian region, if you look at this particular region, mostly cotton is approved in many countries. Cotton is approved in Sudan, Ethiopia, Pakistan. In Bangladesh, it is only one country in South Asia where they have approved GM brinjal, or I can say BT brinjal. But if you look at the Western countries have already given approval to many other crops apart from BT cotton. For example, say US and Canada, you will see that they have given permission to many edible crops, to the maize, to GM potato, to GM peas, GM papaya, GM corn. So many other crops have been given permission in North American continent as well as South American continent. So what is the idea here is to understand that there is more and more acceptance of genetically modified crops, not per se in the South Asian region, but in the other parts of the world, there is more acceptance. So what we can expect going forward is more and more promotion of genetically modified crops even in India. Again, any decision that is taken, it would depend on building consensus among different stakeholders, especially the farmer brethren, right? Farmers, they have to be taken into confidence. Awareness has to be created in the farming community, and then if there are any issues, if there are any protests against this, they have to be catered into before any decision making in our country. All right. Right. So that's about the genetically modified crops, one of the applications, a major application in India. Apart from that, if we talk about the recombinant DNA technology, the evolution, initially we used many nucleases, many ligases, polymerase enzymes, etc. But since recent times, I would say a decade or so, genetic engineering has seen a huge revolution because of the development of new technology called as CRISPR. CRISPR basically stands for clustered regularly interspaced palindromic repeats. Now, there is a term called as repeats. Now, we need to understand how it works. Now, CRISPR is an idea. CRISPR is a technology which is a natural phenomena, which is occurring in a bacteria, and this bacteria is the Streptococcus pyogenes. Streptococcus pyogenes, it developed a self-defense mechanism using something called as CRISPR-Cas9 system. It's a natural system in a bacteria. See, every organism, it aims to survive. Now, bacteria, if it is impacted by other organisms, you do know that viruses can infect bacteria, especially the bacteriophages. They can finish bacteria. Those are the viruses which can end bacteria. So one such virus, it attacked Streptococcus pyogenes. Now, everybody has that instinct for survival. So what this bacteria did, it developed a system of self-defense. Under that, it created, or it encoded a protein called as Cas9. Now, what this Cas9 does for the bacteria is it identifies the genetic code of the virus. It identifies the genetic code of the virus. Every virus, you do understand, there is a unique code, right? If you see a virus, it will have either DNA or RNA, but whatever it is, it will have a specific code which would be repeating in its DNA. You know that. Now, what this bacteria does, it produces Cas9. Cas9 is a protein. Now this protein, what it is going to do, it is going to identify the genetic information of that virus, and it is going to act on it, and it is going to cut that genetic information, not just at one place. That is the meaning of CRISPR. Wherever that genetic information is being repeated in the virus, everywhere that genetic information is cut. That means the virus is dead. Correct? That is a self-defense mechanism. Now, what we did is we have developed an engineered complex of CRISPR-Cas9. Now, we have learned this technology from bacteria. We have, I would say, reverse engineered it. And what we have done is we have created a Cas9, CRISPR-Cas9 system where I send that code, or I can guide the Cas9 protein to cut the double strand of DNA at a specific location, not just at one point, but wherever it is repeating, everywhere it is going to cut. How? I have given it a guide RNA. Now, using a guide RNA, it goes to that sequence of the DNA which I want to cut, and wherever it is present, imagine that this is a sequence of DNA and there is a code. Say I have a code called ACT, right? Adenine, cytosine, thymine. Now I want to cut this particular sequence. Now wherever you will find this sequence, everywhere the DNA would be cut. That means this particular part of your DNA is now edited, and that gene or its characteristics will not be shown at all. That is what we have engineered right now. Now please understand why this CRISPR-Cas9 is novel. Now we already had so many different enzymes, so many other technologies developed for recombinant DNA technology. Like before CRISPR-Cas9, we used different meganucleases. We call them homing meganucleases. We used zinc finger nucleases. We used talon. Talon is another activated nucleus which identifies a code and which can go for a double strand cut. And we also have certain nucleases where we do joining. That means we have ligases, we have different enzymes which can lead to homologous directed repair. Now these are some of the different ideas which we have developed. That means when a DNA is cut, either I allow it to join automatically, that means non-homologous end joining, or I can edit that. So what we have done is, we now, by using CRISPR technology, we have created complexes where I have created an engineered system of CRISPR-Cas9 plus the homologous directed repair mechanism as well. So what it does is it simply breaks the DNA wherever you want, and it adds a different code that you desire. And it has found out that its feasibility is very high. Now, if you look at this, the feasibility of meganucleases, ZFN, TALEN, and CRISPR-Cas9, highest feasibility and highest repeatability and accuracy has come for CRISPR-Cas9. This has revolutionized the genetic engineering domain itself. Now, the question is, sir, where is it being used? Where is it being researched? In so many areas, because now it has higher utility than any sort of other nucleases that we used. It is being researched in almost all the avenues where we use the recombinant DNA technology before. So CRISPR-Cas9 has revolutionized the genetic engineering field, and recently scientists from India, they have created gene-edited mustard. Gene-edited mustard. Now please remember, this is gene-edited, meaning this is not modified. This is not modified. Now, when I modify, that means I have incorporated some other gene. Now, you know, in a genetically modified mustard, I have taken 3 genes from Bacillus amyloliquefacins. I have taken a gene called as Basta, Barnase, and Bar gene, and I have incorporated those genes in mustard and created a hybrid out of it. But now, scientists using CRISPR-Cas9 technology, they have edited Indian variant. Now, Indian variant Varuna, it has some problems. Mainly, it is highly pungent. Why? Because of the presence of glucosinolates. Now, these glucosinolates, they make Indian mustard very pungent, and whatever residue is left out after the after preparing that oil, whatever residue is left out, that residue is used for feed or fodder for animals because it is very protein rich. But it is not highly useful as of now because of the high levels of glucosinolates. To resolve this particular issue, what Indian scientists have done is they have used CRISPR-Cas9 technology. This is the genetically modified organism where I cut one particular part and I incorporate a gene, or even if I don't cut it, when I introduce a new gene, its genes are modified. But right now, what scientists have done is they have simply edited using CRISPR-Cas9, meaning one particular gene which was responsible for high levels of glucosinolate in the seeds, it has been edited. Now, scientists studied that in mustard there are genes called as GTR1 and GTR2. Now they have edited these genes. There are 12 genes of GTR1 6 genes, GTR2 6 genes. Out of that, 10 genes have been edited to ensure that glucosinolates are not present, or less amount of glucosinolates are present in the seeds of mustard going forward. Isn't it revolutionary? This is one of the applications of CRISPR from very recent time, a study which was published, I think, last week or 1 or 2 weeks before. But CRISPR has also revolutionized other fields. For instance, it has revolutionized the idea of stem cell research. Now, stem cell research has gained a big fillip because of CRISPR-Cas9. Let's try to understand stem cell research and some nuances connected to it. The first concept, what is the meaning of stem cell? Stem cell is a cell, it is an advanced cell which has potential to develop into different types of cells in our body. Now, let me give you an example. Stem cell. Now, you do know whenever fertilization takes place. Now, this is the ovum. This is the sperm. When fertilization takes place, you do understand this leads to creation of one nucleus, and this we call it as zygote. Now, I can call zygote to be the most advanced stem cell. It is a totipotent stem cell, meaning a single cell has a capability to divide itself in such a way that it can lead to development of an infant, meaning a single cell now leading to the formation of different organs, skin, formation of brain cells, etc. So stem cell is something, a cell which has a remarkable potential of developing into another kind of cell. Now, where do you find stem cell? Of course, zygote is one of the examples, but it would be rather useless because zygote will undergo cell division very, very quickly. But we have many kinds of stem cells, especially in the embryo, that is the new infant that is being developed inside the womb of a mother. We have stem cells even in adults. You do understand that my bone marrow has stem cells. Some of them can become red blood cells, some of them can become white blood cells, some of them can become platelets, right? So we have stem cells in adult body as well. Now you have a question, sir, stem cell in adults, can we use them for research? Now, stem cell, first of all, stem cell is a special cell which has amazing power to transform into different cells or different tissues of our body. Now, we have adult stem cells, yes, but adult stem cells, they are limited, and that is why there is very limited research also going on adult stem cells. Why? Because adult stem cells, I wouldn't call them totipotent. Of course, totipotent is zygote. I wouldn't call them pluripotent as well, meaning their ability to transform into different cells is rather less. Yes, they are multipotent. For example, stem cells in my bone marrow, they have capability to turn into, say, 5 to 6 kinds of different cells, probably 10 different cells. But what I want is a stem cell which is pluripotent, and I can find those stem cells where? In the embryonic stem cells. And this embryonic stem cells, where can they be found? They can be found, or they can be retrieved from umbilical cord, or they can also be retrieved from the amniotic fluid. The amniotic fluid, when the baby is being born, you can take this, yes. But again, embryonic stem cell research, for creating an embryo just for research, it leads to some of the ethical issues which are already plaguing the biotechnology industry. Moreover, adult stem cells, they cannot be used because of the potential problem of pollution, potential problem of say gene mutation. Adult stem cells, uh, they're not pristine, they're not pure. The best kind of stem cell on which we can research is embryonic stem cells. But here, the idea of ethical dilemma does come in between. Meaning, for my research, I have to develop an embryo, I extract stem cell from it, and then what of that embryo? The right to life activists, they suggest that life begins with embryo. So embryo also has rights. That is where this catch or this dilemma comes in. And hence, the scientists started to develop an idea of induced pluripotency. Meaning, I take a simple somatic cell from a healthy adult whose cells are, say, healthy, they are quite pure, they're not adulterated, they do not have any mutation, etc. Take a somatic cell and induce pluripotency in it. From 2007, scientists have gained this expertise. In fact, 2007, if I'm not wrong, Nobel Prize was given for induced pluripotency itself to some Japanese scientists. So induced pluripotency is an idea that is gaining a lot of traction. And for that, from the last 10 to 15 years, scientists have come up with different ideas. Say, I can induce pluripotency by taking a stem cell and by transferring the nucleus of that stem cell. You do understand that pluripotency is because of specific information in the nucleus of a stem cell, correct? So what scientists did was they transferred the nucleus from a stem cell into the normal somatic cell. So, okay, maybe we can induce pluripotency. They came up with other ideas of cell fusion, fusing somatic cell with the stem cell. They also came up with other ideas of transcription and transduction or small molecule compound based induction, where pluripotency was induced. Again, these are all very complex processes which led to induced pluripotency. But today we have another idea. Called as CRISPR. Now this CRISPR technology, what can it do? Imagine its potential that I take a somatic cell. I already have nucleus here, and in this nucleus there is nuclear information, and that is why this cell's ability is very limited. Now, what I need to study the information or the genetic information in the stem cell, whatever stem cell, the most advanced pluripotent cell. I take genetic information from that and I study those genes which are responsible for pluripotency, and I incorporate only those genes in the somatic cell. So don't you think the somatic cell will become a pluripotent cell? Yes, and that is why stem cell based research has become a game changer for developing induced pluripotency stem cells by avoiding the ethical dilemmas that are connected with embryonic research, and by eliminating this issue of limited availability of adult stem cells. So I don't need to use adult stem cells. I need to use simple adult somatic cells and induce pluripotency in them to turn them into stem cells. Quite extraordinary, all right? And one of the biggest applications of this is the regenerative medicine. Now imagine, under regenerative medicine, what scientists are doing, if you have problem with one particular organ, simply take the adult somatic cells, induce pluripotency in them so that they become stem cells, pluripotent stem cells, and give this stem cell as a treatment for regenerating that particular organ, that particular tissue, or that particular part. Quite extraordinary, isn't it? And in recent times, CRISPR-Cas9 is also being used in immunotherapy. Now, CRISPR and CAR T cell therapy, you must have heard. This is one of the recent applications. Now, what the scientists have done to fight cancer, one of the last method or last treatment lines is the immunotherapy treatment. Now, under the immunotherapy treatment, T cells, the T cells which are a part and parcel of our immune system, these T cells are taken, and these T cells are edited using CRISPR-Cas9 so that there is a specific protein called as chimeric antigen receptor protein which is induced on the T cells. And these genetically edited T cells, they are injected back to the patient. Now the CAR T cells, chimeric antigen receptor T cells, they will find cancer cells and they'll finish these cancer cells. This is one of the lines of treatment which has been evolved. Now this is a method. What do? The healthcare providers, they collect the blood cells, collect the blood, and from that blood they separate the T cells and send back the original blood. So these T cells are taken, and in these T cells, using CRISPR-Cas9 technology, a gene is introduced, a new gene is introduced in the T cells. Now through this new gene, what happens is these T cells, they will develop a chimeric antigen receptor protein on them. And once it is done, these cells are cultured. And after culturing them, you will find millions of CAR T cells. And these CAR T cells are now introduced inside the patient. And these CAR T cells will show their magic. They are now activated cells which can identify cancer cells and they can finish cancer cells. If this research is successful, in fact, there are many hospitals even in India which are providing this particular treatment. This is going to be quite extraordinary. Right now, the biggest problem is, I would say, the cost constraint and the efficacy part. There is a lot of research going on in this particular area. We'll keep an eye on that. But from exam perspective, there might be some questions because previously we have seen stem cell based questions in mains examination where question was asked that can stem cell therapy be used to even treat genetic disorders like say thalassemia, etc. So on those lines, some questions have appeared. So do have this knowledge about CAR T as well. Now, apart from that, there is one more topic. I wouldn't say completely connected with CRISPR-Cas9, but it is connected with genetic engineering. It is called as mitochondrial replacement therapy. It is not connected with recombinant DNA. To some extent, yes, but it is not connected with CRISPR for sure. Mitochondrial replacement therapy. Now, this is in news from UK, where recently 3 parent babies have been developed to eliminate mitochondrial disorders, mitochondria based disorders. Now, mitochondrial DNA disorders are quite rare. 1 in 5,000 or 1 in 6,000 patients have mitochondrial DNA issues. And whenever there is an issue with mitochondrial DNA, you do understand that the patient will have or will be suffering from some sort of weakness there, because mitochondria is connected with the energy in the body. It is called the powerhouse of a cell. And if the mitochondria is problematic, then the entire energy system of your body is problematic. So they might have problem with their muscles, problem with their heart, problem with any of the organs, problem with eyesight, problem with bones, basically anything connected with energy, or problems connected with energy, problems connected with fatigue can be created because of mitochondrial DNA disorders. Now, these are the disorders which are inherited. There is no treatment available to it. As of now. Why? Because it is a genetic disorder. And how is it inherited? Please remember, mitochondrial DNA is only inherited from mother sides. Every individual in this world, they have mitochondrial DNA from their mother side only. Why? Because see, this is the ovum which comes from female. Now this ovum has 23 chromosomes in this nucleus, and they also have mitochondria. Let me draw this mitochondria using a different color so that you understand. This is the nucleus, and I'll draw this mitochondria in black. Now this is a problematic mitochondria from a female. Now, when a sperm fertilizes this, please remember, when a sperm fertilizes this, sperm has its own nucleus, and the mitochondria of the sperm is outside the cell body. So as soon as it fertilizes, then this tail, it is discarded. Now, since this tail is discarded, that means the male mitochondria, it does not enter the ovum at all. The nucleus from the sperm, it is going to fertilize this. Now this is going to have 46 chromosomes, which basically translate to about 25,000 genes in humans. But the offspring or the individual, the child, it will get mitochondria only from mother's side. Now this mitochondria, you do know it has a circular DNA, and it translates or it encodes 37 genes, and these genes are mostly connected with your energy requirement of your body. Now imagine if the mother's mitochondria is defective, there is no solution to it. It's a genetic disorder. So how can we resolve this issue? Now for this, new treatment methods have come, which is called as mitochondrial replacement therapy, where the mother's ovum, you will see mother's ovum. Now this ovum has mother's nuclear information, and this is the bad mitochondria which the mother's ovum has. So what scientists have done is they have enucleated this nucleus. That means nucleus from the mother is taken. That means the main DNA that is there, it is already taken. Another donor is called a donor's egg is now taken, and a donor's egg, its nucleus is removed. So enucleation from donor's egg is proceeded with. Now this donor's egg, it has donor's mitochondria. Basically this donor's egg or donor's ovum, it has everything except for the nucleus, and this nucleus from the mother's side, it is now incorporated in the donor's egg, and this egg is then fertilized using the father's sperm. What is going to happen now? The nuclear information which comes, it has come from two parents, but the egg itself is derived from another donor. But what will happen? The offspring that is born, it is going to have genetic information of the natural mother and father, and also 37 genes or genetic information even from the donor. That is the idea of three parent babies. But using this technique, mitochondrial DNA disorders have been resolved. Yes, again there are other methods as well. For instance, we can take a directly fertilized egg as well. For instance, mother's egg is taken, mother's egg is fertilized with the father's sperm, a zygote is formed, and zygote's nucleus can be taken out. But again, as I told you, dealing with zygote will be a little tricky because it undergoes cell division very quickly. So this is the method that is used standardly. Just remove the egg from the remove the egg from mother's side, denucleate it. That means remove the nucleus, add that nucleus to donor cell. By removing this donor's nucleus, fertilize it. That's the mitochondrial replacement therapy. Now throughout our interaction, I have been telling very lightly, or I'd say in a very easy way, remove the nucleus, do these things, cut the DNA, join the DNA, all these things. Yes, from academic perspective, we are learning the basic technique. We are not supposed to get too much into the technicalities, but whenever I say these terms so easily, there is effort of scientists which has gone into it. And although I'm giving you the gist of it, the theme of the treatment method, but it takes a lot of time and it is highly sophisticated as well. Just for your understanding, I to put it in simple terms, I told in that way. All right. So these are some of the important things or important facets which we need to know about genetic engineering. The questions might come out on this. The questions might come out on what do you understand by mitochondrial replacement therapy. And in such questions, if you're running out of time, you can always use the diagrams. Again, not highly technical diagrams as well, just to explain that you know the process. Such ideas can be used very easily. All right. So genetic engineering and related topics, I would like you to focus specifically on MRT, CAR T therapy, CRISPR-Cas9 and applications of CRISPR-Cas9, and genetically modified mustard to some extent. All right. Right. So prepare very well. All the very best. We'll meet in the next session. And if you like this video, you can always follow me on this particular ID at Rahul Sai 222. If you have any doubts, any queries regarding preparation, you can always reach out to me on IG. Thank you for watching this video. Jai Hind.